Hello, and welcome to our global HMO webinar hosted by dsm-firmenich. My name is Taichi Inui. I am the moderator and the host for this webinar. We have two speakers today, Professor Sharon Donovan, who is Professor and a Melissa M. Noel Endowed Chair in Nutrition and Health at University of Illinois at Urbana-Champaign. We also have James Young, who is Vice President in Early Life Nutrition at the dsm-firmenich. Sharon will present us with the latest scientific studies in HMO. James will talk about global market trend of HMO and development around them at dsm-firmenich. Before we get started, if you have questions, please submit them through the Q&A engagement tool, which is located below the video screen. Any questions that we don't have time to respond during today's session, we will address via email following the presentation. Also, the recording will be available on demand on nutritioninsight.com, as well as our own website on dsm.com. A link to the on-demand webinar will be emailed to you after today's session. With this, I would like to hand over to our first speaker, James Young. James, the floor is yours.
Thanks very much, Taichi, and hello everyone. Nice to see everybody. I want to take us through a few market trends that we're seeing with respect to human milk oligosaccharides in the infant formula market. So first of all, I'd like to say that infant formula launches with HMOs continue to grow. In fact, in 2023, external data that we see shows that roughly 20% of launches in the infant formula market and the follow-on formula market contained HMOs. Since their launch in 2018, we've seen a number of trends and drivers happening in this market. First of all, there have been a lot more infant formula players. From the early big global players, we're now seeing lots of local players getting into this market to try and mimic the nutritional composition of breast milk as closely as possible by adding HMOs in their formulas. We've also started to see the move of HMOs from mainly premium products when they launched into slightly lower down the brand penetration into less premium segments, and we fully expect that trend to continue. We're also seeing new HMOs at current dosages changing. Nestle and Abbott currently are the only two players to have launched multiple HMO products, but other main players are expected to follow. We're seeing lots of ideas around 5 HMOs and beyond in the market. We're also seeing a change in dosage philosophy. Some of the low dosage markets are expected to harmonize towards high dosage markets over time, as benefits from the higher dosage of HMOs, particularly getting them closer to physiological levels, are supported by further clinical trial evidence. And last but not least, we're seeing increasing regulatory approval in new HMO markets. Of particular excitement is both 2FL and LNNT recently getting approval in China, and we expect further regulatory approvals in many new markets to expand the addressable market. I'd now like to take us through a little bit about dsm-firmenich capabilities in HMOs. dsm-firmenich offers a unique expertise and capabilities to support your needs. As an HMO market maker, we have a dedicated team of HMO scientists committed to expanding HMO knowledge around the world. One of the things we're most proud of is our HMO donation program. So whilst we obviously commercialize HMOs, we're very committed to science building as well. And we have our HMO donation program to help academics and other academic institutes do further research on HMOs. We have more than 25 HMO structures available and many mixtures of HMOs as well for donation. And we are currently supporting over 100 projects across the world, which have resulted so far in over 50 publications. We're also very proud of the review paper we created a few years ago on the mean of milk, which is a review of human milk oligosaccharide concentrations throughout lactation and has now been cited over 100 times in a variety of different journals. We have a suite of tools designed to support you in your new product development and a team of formulation experts and application specialists who can help you formulate with HMOs. We also have access to an extensive nutritional product portfolio across early life nutrition, Not just HMOs, but we are industry leaders in lipids and vitamins and many other things as well. We also have created a new preclinical lab dedicated to HMO research, and we can offer 8 HMOs as individual ingredients or straights, but also as HMO blends, as combinations or premixes with other ingredients, depending on what your customers' needs are. dsm-firmenich HMOs are the most widely clinically tested HMOs available on the market, and we're very proud to have the broadest breadth of regulatory approval of anybody in the market. We also have a very strong advocacy team who support our regulatory teams and our customers in helping HMOs and HMO science. In terms of our global reach, as I mentioned, we have a rich portfolio of 8 HMOs today, with more coming, and it covers all 3 structural HMO classes. We currently have the leading regulatory position across the world with 90 explicit HMO approvals and HMOs that can be commercialized in over 165 countries. We have 8 HMOs approved in the US, soon also in the EU and UK, and 5 or 6 HMOs approved in the rest of the world. dsm-firmenich is the only HMO manufacturer that succeeded with the approval for 2 HMOs in China, with 2FL and LNNT being approved late last year, and we expect to see those in infant formulas later this year. dsm-firmenich is leading in regulatory market access worldwide with over 100 explicit HMO approvals, and we're very proud to continue that progress with our customer base. We're also very proud of our manufacturing base. DSM has an exclusive plant based in Esbjerg in Denmark, which is dedicated to HMO production. It's the world's largest fully integrated HMO production facility, and although it was originally built as a milk powder factory and later was modified as an amino acid before we acquired it, after a 2-year rebuild, it is now a state-of-the-art facility supplying all of our HMO needs. It has a proven track record of over 5 years of uninterrupted supply, and there's ample room for significant upsizing. The facility is prepared and being designed in a modular way so that we can expand as we need to. We also are very proud that we're able to provide both dry and wet mix compliant HMOs across our portfolio. Now back to Tai Chi, who will introduce Sharon Donovan.
Thank you, James. That was quite insightful talk. Now I would like to move to our next speaker, Professor Sharon Donovan, who will talk about the latest scientific development of HMO. Sharon, floor is yours.
Hello, my name is Sharon Donovan. I'm a professor at the University of Illinois in Urbana-Champaign, and it's my pleasure today to present to you on the evidence for benefits of human milk oligosaccharides in supporting infant development. So for the next 30 minutes, I'll be presenting some background on the structure of HMOs, their concentrations and variation in human milk, their biological activities, and then I'll give a summary of the findings from randomized placebo-controlled clinical interventions, both HMO supplementation and infant formula. We'll touch on some of the potential mechanisms and then finish up with some conclusions and future opportunities. So this slide shows a comparison of human milk oligosaccharides between human and bovine milk. And so you see on the left, the blue box indicates the milk oligosaccharides in human milk versus bovine milk. And one thing that's quite apparent is that bovine milk contains much less overall HMOs. And since most infant formulas are made with bovine milk, we're starting with a material that has a low level of oligosaccharides. In addition, we also see that the structural complexity of bovine milk is much lower than human milk. So to summarize, some of the unique aspects of human milk is that human milk is higher in total HMO concentration. It has a much greater diversity. Over 200 different structures have been identified. Most are not found in other milks, and about 22 of these HMOs account for more than 90% of the total HMO content. So even though it seems daunting that there's over 200 forms, it's a smaller percentage that we can look at as potential targets for HMO supplementation. Another unique aspect of human milk is that the human milk oligosaccharides are highly fucosylated, and that is much different than any other species, including bovine milk. So on the left, we see the structural components of HMOs. So we have 5 core monosaccharides that are used to make HMOs. HMOs are synthesized in the mammary gland. And they all start with lactose, and then they're formed by elongation and or fucosylation or silylation. And so basically we have this initial lactose, which is built upon to get this structural diversity. And these glycosidic bonds that are formed in making HMOs are non-digestible by mammals. And so I oftentimes refer to HMOs as sort of the fiber of human milk. So what that means is that they can pass through the infant digestive tract and not be broken down. However, when they get into the colon, then we can start to see some bacteria begin to utilize those HMOs. And we'll talk more about that later. So this slide shows a summary of the major HMOs in human milk of secretor mothers, and I'll explain what that means. And so you can see this orange wedge here is 2-fucosylactose, and then you see many other HMOs that have fucose bound to them. They're just longer products. We also see a neutral or non-fucosylated or silated HMO, and then we see some of the silated ones. And again, these are the predominant ones in human milk. So what factors affect human milk concentrate or HMOs in human milk? One is stage of lactation. We generally see that total HMO content declines over time, but when we look at individual HMOs, some go down and some go up. And predominantly the fucosylated ones are declining, but we actually see some silylated HMOs increase over lactation. I think the unique part of human milk oligosaccharides is not every woman secretes the same HMOs. And one of the key factors that influences HMO composition is basically the presence, the expression of glycosyl transferases. So the alpha-1,2 fucosyl transferase, which is known as the FUT2 or secretor gene, is very significant in influencing HMO concentration. So as I showed you on the previous slide, in secretor mothers, 2FL is actually one of the predominant HMOs, but that is not present in the milk of non-secretors. We also have the FUT3 gene or the Lewis gene, which produces the 3-fucosylated HMOs. So this is really the major factor. There's a number of studies that have looked at other factors in the environment or diet, and they're not completely consistent. There may be some effects, but they're much less than the genetic aspect. So this slide summarizes the difference between HMO total content and composition between a non-secretor and a secretor mom. So what you see is that the secretor mothers have a much higher concentration of total HMOs, about 15.9 versus around 9. And again, this is predominantly due to lower levels of the 2FL or the absence of 2FL in non-secretors. So it's not just the 2FL, it's also these elongation products that can be fucosylated. So then we see a greater diversity of HMOs in secretors or lower diversity in non-secretors. So one of the interesting aspects of this secretor status is that it's not the same and that we see global variations. And on average, about 80% of women are secretors and 20% are non-secretors. But as we look around the world, we see that secretor status is higher in mothers in South America, whereas in Bolivia, 100% of the women tested are secretors, and similar high levels in Peru, Brazil, and Argentina. Whereas when we look in Africa, we see variation across the continent, but we see more in the 60 to 75% of women being secretors. So there's quite a bit of global variation, and it's thought that this was selected for during evolution. I'll talk, one of the things that this fucosal lactose can do is to inhibit binding of pathogens to the lining of the gastrointestinal tract, and so that the presence of endemic pathogens may have provided selective pressure towards keeping this genetic variation at a higher or lower level. So you can see there isn't a lot of data from this paper in Chinese populations. So I've added another table here from 3 different recent papers in looking at secretor status in the Chinese population. And overall, you can see around 75 to 80%. So again, remember, worldwide, the average is about 80%. So I would say that the Chinese population is similar to the average. So now let's turn to some of the biological activities of HMOs. And so one of the big questions is, well, why does human milk have such high concentrations of HMO? It's the 3rd most predominant solid component. So that's led to a lot of research on potential biological activities. Some of these data's come from in vitro studies, some from preclinical or clinical studies, as well as epidemiological studies, again, because we have populations where we can compare infants nursing from secretor versus non-secretor moms. We can start to look, are there health benefits, for example, even within the context of human milk, of the fucosylated HMOs. So again, these HMOs affect numerous glycan-mediated processes, such as microbial colonization, development of the immune system, infectivity of pathogens, and as well as neurocognitive development. And we know that very small amounts of HMOs and their microbial metabolites can enter the circulation and may have systemic effects. So we see that HMOs have been implicated in gut development, reducing the risks of infections, and I'll show more of that on the next slide, modulating mucosal and systemic immunity, influencing neurocognitive development. Work in my lab has been looking at this in the piglet model. Microbiome structure and function. We know that some microbes, particularly the Bifidobacterium longum infantis strain, has a specific gene cassette that allows it to utilize HMOs quite effectively and even outcompete other bifidobacterial strains. There's some evidence of growth in anthropometry. Again, most of this has come from epidemiological studies where they've looked at differences in HMO concentrations in human milk. As I'll show you, in studies where we actually add HMOs to infant formulas, there has not been any demonstrated differences in growth. And one of the newer areas, since we know that it can affect neurocognitive development as well as microbiome structure, is potentially having these HMOs work through the microbiome gut-brain axis. So this is a table adapted from a review article by Springer et al. And it's basically, I've pointed out the reduced risk of immune-related outcomes in human infants, because really I think this is the strongest evidence. And so some of these are things like necrotizing enterocolitis. You also see eczema, allergy, immune sensitization. We also can see lots of benefits for inhibiting infectious diseases. So it's thought that some of the benefits that we see of human milk feeding are in part due to the presence of HMOs. Obviously, there's other immune components in human milk, but human milk as a package provides many components to help the infants resist infections. As you can see, there's a variety of HMOs that have been implicated, but you predominantly see a lot of these are the fucosylated ones. Most are from observational trials, but some are from RCTs. So now let's transition into some of the studies where we've actually investigated if we add specific HMOs to infant formula, do we see differences compared to unsupplemented formula? And in many but not all studies, there's also a breastfed reference control. So, where are we? I've said there's hundreds of HMOs. We know most women, have about 22. Currently, there's 8 HMOs that have been received either GRAS status in the U.S. or have received a positive opinion. 7 have received positive opinions from EFSA. And so, we're getting more HMOs available, and I'll show you that in some of the clinical studies. 4 have been approved by the Food Safety Agency of Australia and New Zealand, and 2 in China. Again, as I'll show you, most of the early studies only supplemented 1 or 2 infant formulas, but we're now starting to see clinical studies and even commercial products that have 5 to 6. So as countries approve more HMOs, you can expect likely to start to see more HMOs in the infant formulas in those countries. So I'm going to refer you to this recent systematic review of the clinical studies on the supplementation of manufactured HMOs. I'm just going to touch on some of this, but if you're interested in the overall outcomes as well as some applications beyond infancy, I'm really going to focus on the studies in term infants, but there's some studies with infants with medical indications, older children as well as adults. And these studies they reviewed a total of 31 studies. 26 were the actual initial clinical trial, and 5 were publications of follow-up studies for future analysis of the data generated in the clinical trial. These studies included 8 different HMO structures, either individually as blends, and some with either pre or probiotics, but I'm not going to talk as much about the probiotics today. And all trials, many of them were simply that first step of safety and tolerance assessment, but others also assessed efficacy outcomes such as stool characteristics, infections, gut microbiome, etc. So this is a summary of the main HMOs that have been conducted in healthy term infants, and these are all concentrations or doses in grams per liter. You can see the reference and the year that it was conducted. So there was a very early one by Prieto with just LNNT. Probably the next sort of bunch of papers that were published were around 2017, 2015, 2016. So these were either looking at 2FL and LNNT or just 2FL alone. As you can see, the papers now coming out in 21, 22, not only are we starting to see some of the higher concentrations, but we're seeing studies where they're including 5 to 6 HMOs. And I've put down here the reference level for human milk. So you can see even though some of the recent papers are now at around 5.75 grams per liter, that's still only about half of what a typical breastfed infant would be consuming in sort of term human milk. But again, we're getting close to the range of 2FL exposure and 3FL. So again, we're getting there. A lot of these papers did not include any other prebiotic. Others having put the HMOs in, but also within the context of the prebiotic that was already present in that infant formula. So I want to just touch on why variety and dose is important. Again, some of the commercial infant formulas right now still only have 1 HMO in that. And obviously, these HMOs can have different functions in the body. And first of all, it's difficult to get to a concentration that's in human milk. So again, most of the infant trials, early ones, were only about 2 to 10% of the levels in human milk. And there were few of the studies until recently had actually looked at any of the silylated HMOs that have been focused primarily on fucosylated. And we're beginning to understand that silylated HMOs likely have very important functions for the infant as well. And, we've talked a lot about the fucosylated and bifidobacteria, but now there's been a couple of recent papers suggesting that these silylated HMOs can promote the growth of Bacteroides species in infants. And so Bacteroides are also a predominant HMO or predominant microbe in the infant. They also, many pathogens bind to sialic acid on cell membranes, and so the silolactoses can inhibit some of that antiviral or bacteriostatic activities by adding, basically hindering the binding of pathogens to the mucosal lining. And some preclinical studies where they could actually knock out the gene in mice, they found that when mice suckled dams who did not have sialylated HMOs in the milk, they could see differences in nervous system development and neuronal patterns. And so that was suggesting some role also in neurocognitive development. So, as we move forward, we would like to see a more complex complement of HMOs in infant formulas, and also in concentrations that are more closely approaching that of human milk. So I'm just going to give you an snapshot of the overall outcomes. And so by and large, really every study has shown that HMO supplementation at the levels tested is safe and well tolerated. No differences in growth parameters, no differences in tolerance, so things like gassiness, stool, spitting up, stool consistency, particularly at lower doses. No differences in adverse effects or serious adverse effects, and also no reported differences in sleep or behavioral outcomes. As we, some of the studies, particularly those that are approaching higher concentrations, are beginning to show some functional outcomes. And in many cases, these can either be significantly different from the unsupplemented formula, and also can be shifting them towards breastfed infants. So in most cases, as I'll show you, they're kind of shifted to an intermediate between the breastfed infant and the infants fed the unsupplemented formula. So these can include some of the softer stool characteristics, changes in gut microbiome composition, not only promoting healthier bacteria, but also reducing some potential pathogens. And immune markers have been reported in the blood as well as in the stool. And in the Puccio study, they had a reduction in parent-reported adverse health outcomes. So fewer days with fever, less use of antipyretics or anti-fever medication, etc. So there seems to be, no concerns about safety, and some indication that there may be some benefit to the infant. So now I'm going to take a little bit of a deeper dive into a recent paper from Boshiva et al. And the reason we're focusing on this is a 5 HMO supplementation study, but it's also one of the largest HMO RCTs, and actually one of the largest infant RCTs. So there were 693 infants. There were 3 randomized formula-fed groups, so they tested HMOs at 2 doses, and they included a human milk reference group. So these were the HMOs that they studied. So you can see 2FL, difucosyl lactose, lacto and tetrose, 3 silolactose, and 6 silolactose. So you see some sialylated, some fucosylated, and what we refer to as a neutral HMO. It was also one of the longest studies in terms of being 15 months. And because it was so long, they actually were able to compare or look over 3 different stages of formula. So a standard infant formula for the first 6 months, a follow-up formula for the second 6 months, and then a follow-up milk from 12 to 15 months. So it's a quite extensive paper. What was presented in the Boshiva paper, though, is just the first stage, the enrollment to 6 months. So this was a multi-center study in 32 sites in Bulgaria, Hungary, and Poland. They enrolled healthy term infants between 1 and 3 weeks of age. And again, they were randomized into 3 formula-fed groups, and then they also enrolled a non-randomized human milk reference group. So the control group, what we will refer to as test group 1, contained 1.5 grams per liter total HMOs, and test group 2 contained 2.5 grams per liter HMOs. So what they're reporting in this paper is focusing on some of the fecal microbiome characteristics. So they looked at shotgun metagenomics for this study, and they also looked at some of the metabolites and biomarkers. And so we'll go through a couple slides of the actual data. And what this shows is a principal coordinates analysis of the microbiota. So this is giving you sort of an overview. Each dot indicates an individual infant. And what we're looking at at baseline, so the yellow group is the human milk group, and then these are the 3 formula groups. And so again, at baseline, at enrollment, there was no difference. By 3 months of age, you can start to see the groups, the formula-fed groups, moving a little bit closer to the human milk group. But by 6 months of age, so the green group is the control formula, and these are the 2 HMO supplemented formulas. So you can actually see that those 2 groups, their overall microbiota composition is now shifted to be much closer to those infants fed human milk and significantly different from those infants fed the unsupplemented infant formula. So it's a gradual process, but by 6 months of age, those infants receiving the HMOs at either dose, 1.5 or 2.5, looked more, in terms of their microbiome composition, more like the human milk than the formula. So there's a number of figures in this paper. I've just pulled out a few. This is bifidobacterium, again at baseline, 3 months of age and 6 months of age. So you can see at baseline, all the formula groups are different from human milk. And as we move through by 6 months of age, you can see that the human milk is different from the formula, but it's not significantly different from either of the HMO supplemented groups. So that's saying not only is the overall microbiome composition shifting, but when we look specifically at bifidobacteria, we see that its relative abundance has also been shifted to look more like human milk. And again, bifidobacteria is a healthy bacteria and one that we want to promote in infants. They also looked at the relative abundance of what they would say would be potential pathogens. So again, in this case, C. difficile. So again, there wasn't really any difference at baseline, but you can see at 3 and 6 months of age that the 2 groups that were receiving the HMO are lower and sort of intermediate with the human milk group. But the significant difference is that the relative abundance of C. difficile is lower in these 2 groups compared to formula alone. So it's not only promoting good bacteria, but it seems to be suppressing pathogens that we don't want to be seeing in the infant gut. They also looked at some other markers of intestinal immunity. So again, the human milk group in the yellow and the control formula, and at 3 months, you can see that the 2 HMO groups are significantly different from control. And at 6 months, the higher dose HMO was also significantly different from control. But they're both, lower than the breastfed infant. But, keep in mind that human milk contains secretory IgA. So when we measure that in the stool, it could reflect not only the IgA in the milk of the mother's milk, but also what's being produced in the gut. We see a similar trend at 3 months where the amount of alpha-1 antitrypsin is lower in the 2 groups getting the HMO compared to the formula. And then I think I'll just bring those 2 in. So they also looked at some other fecal characteristics. And so what you see is a very nice dose-dependent sort of relationship between fecal pH. And so as components are being fermented in the gut, you can produce organic acids. So some of those are short-chain fatty acids, some are lactate. And so those components are going to reduce the pH. And it's been shown in many studies that the fecal pH of breastfed infants is lower than that of formula-fed infants. And, we're seeing this downward trend there as well. And again, we see with lactate that the two HMO groups are intermediate to the, and still significantly different, but significantly different than the control. And again, these are the specific short-chain fatty acids that we're interested in. And again, you can see that, there's some shifts over time, but these are tending to be intermediate between the breastfed and the human milk or the control formula. So I just have a couple more slides, and this is an analysis of one of the older studies. And this is one thing that's kind of neat, is that we have these samples from these studies that were even conducted back in 2017, and we can sort of reanalyze those samples and look for mechanisms of action. So this was the Puccio study, where they compared two different doses of HMO at a 1.5 grams per liter, so similar to the lower dose in the Boshiva study. And two observations they observed from this study, again, that produced parent-reported bronchitis and lower respiratory tract infections. They also found modified fecal microbiome and metabolites, so similar to the study I just showed you, higher bifidobacteria and acetate. So what they did is they applied a statistical approach called a sparse partial least squares discriminant analysis to see if they could link the sort of microbiome characteristics with the clinical outcomes. So what they found, first of all, is that they could separate what they're calling the cases, which were infants with lower respiratory tract infection, than from the controls. So they were able to statistically separate. And then the next step is to say, okay, well, what are some of the factors that are causing these groups to pull apart? And what they found when they looked at the controls, that bifidobacterium abundance and some of the percent acetate, and also the presence, the product, so basically having the test formula and the fucosal groups, were the factors that were most strongly pulling the two groups apart. And so they then went and looked a little bit more closely. And so again, these are the cases. These are the infants who had lower respiratory tract infection. They're actually seeing it's significantly lower fecal acetate concentration, as well as a lower relative abundance of B. longum species. And this shows a nice correlation between bifidobacterium abundance and fecal acetate, suggesting again that the bifidobacterium is actually producing the acetate. And then they looked at sort of the specificity. So they see a relationship or correlation of all bifidobacteria, but the relationships were really strongest with the B. longum and the B. longum infantis, and not as much with other strains of bifidobacteria you see towards the bottom. So again, it's really using statistical approaches to say, hey, we have this clinical observation and we have these sort of functional outcomes in the gut. Can we start to link these together? So really kind of suggesting that HMOs can promote bifidobacteria, which in turn can ferment the bifidobacteria in turn ferment those HMOs, producing acetate, and those, at least statistically, are contributing to the reduction in the clinical symptoms. So just some quick conclusions, and where do we go next? So we see that these HMOs are multifunctional components in human milk. They affect multiple aspects of infant development. Supplementation of HMO to term infant formula is achievable and safe. And basically in the past, the biggest limitation has just been the availability of these HMOs, but we're now starting to see companies such as DSM producing these in large enough quantities that they can be supplemented to formulas. We see that they alter the microbiota and microbial metabolism, and those, again, are linked to health outcomes in the infant. There's likely other mechanisms, but initially these are some observations. And so, where do we go from here? We still need to know more about mechanisms of action, and this can be preclinical studies, clinical studies, or even using AI on large data sets to try to look at associations which might not be initially intuitive to us. More research on HMO and neurocognitive outcomes. There's strong animal model research, but less in human infants. And now that more infants are consuming formulas with HMOs, it'd be really interesting to look if we're seeing effects on neurocognitive development. Another really hot area is supplementation of preterm infant formula and other specialized formulas. Obviously, preterm infants can have a significant dysbiosis and also risk of necrotizing enterocolitis, so they may be able to benefit from the HMOs. And then this area is actually moving in terms of applications beyond infancy. And if you go back and look at the review article of the RCTs, you'll see some of the work that has been published already in children and adults. But this is really an area that's rapidly expanding. So thank you very much for your attention. I hope you enjoyed the presentation, and I look forward to your questions.
Thank you, Sharon and then James, for your insightful talks. Now let's move on to Q&A section. We have some good questions coming in, and I would like to invite both speakers back in live. And then Sharon, thank you so much for joining in such an early or late hours in your local time. First question, that there are several questions that are overlapping to each other, so I'm trying to combine them and preserve the essence to it. The first question is about the single HMO versus multiple HMOs. Why do we need more HMOs in infant formula when there is already one present? Is there a synergy between HMOs important for the functions and effects? I would like to ask this question to both speakers, but first, Sharon, please share your opinions, please.
Hey, thanks. Yeah, that's a great question. As I presented in my talk, human milk is the gold standard. It's what we want to be getting as close to as possible. And human milk does contain a lot of HMOs and a large variety of HMOs. So any movement that we can make towards getting closer to the concentration and the structural variation would, again, get us closer to the gold standard. And I think at the very least, we do want to try to represent the different forms of HMO. So again, 2FL is great, but as I mentioned, some of the silated HMOs can have their own specific functions, and even the neutral HMOs like the LNT, LNNT are utilized by microbes as a fuel source, so they can be fermented and promote the growth of bifidobacteria and healthy bacteria. So again, if we want to get closer to human milk, we know that we need to be getting a better variety of HMOs. The optimal concentration has not been worked out yet, but as I showed you that at clinical trials where we've provided higher doses, we do tend to start to see more functional outcomes without compromising any sort of safety or tolerance aspects.
Thank you, Sharon. James?
Yeah, I mean, not a lot to add to what Sharon said. I mean, from my perspective, at dsm-firmenich, our curiosity really is driven by the complexity of the HMO profile in human breast milk. Within our early life nutrition business, we very much are driven by giving the best, providing the best start in life for all infants. And we believe getting infant formula as close to breast milk as possible is the right thing to do. As Sharon said, there's somewhere over 200 different structures that have been identified. There must be a reason for that number. Perhaps not all of them are relevant, but we are pretty committed to trying to get as many of them as possible characterized and synthesized. And I think over time, it would be great to see, a wide variety of these different materials, both the fucosylated, adesylated, and the neutral core ones being used across multiple formulas. So we believe that there's a lot to go here, a lot to go for.
Excellent. Thank you. We have next question again about comparing HMO and other available
fibers. So some questions around this. Is HMO viewed as a dietary fiber? What's the difference between HMOs and FOS and NGOS, especially considering there is a price for HMOs significantly higher than GOS and FOS? What are the benefits of HMO compared to FOS and GOS? Again, I would like to ask this to both speakers, and then perhaps this time I will ask you, James, first.
Sure. So I guess there's sort of two questions within here, which is one, the sort of differences between FOS and GOS and HMOs, and then of course the price gap that is real in the market. I think first of all, I would say, again, coming back to getting as close as possible to breast milk, we see, HMOs are a significant proportion of human breast milk. And so they, having human identical versions of those in your infant formulation brings you closer to breast milk, whereas FOS or GOS are not as prevalent by any stretch in infant formula. So, I really don't see a sort of philosophical argument as to why you would not want to put HMOs into infant formula. I think acknowledging the price trend, yes, there is definitely a price gap between FOS and GOS and HMOs. I think it's clear to say, HMOs are still in the infancy of their development. And I think ourselves at dsm-firmenich, as well as our friends in our competitor organizations as well, are working hard to bring the price of those down. And over the last few years, we've seen significant drops in the price. And we are committed to continuing to bring that price down. Whether we ever get it down as close as FOS and GOS, I think is questionable. But I think certainly coupled with the benefits that we are also starting to see through science, I think it makes sense that HMOs are the lead ingredient that you would look to put into your infant formula.
Thanks, James. Sharon, could you provide, from a scientific point of view, the differences between HMO and FOS and GOS or other dietary fibers?
Yeah, so no human milk sample contains FOS, and there may be some galactosyl, like 3-galactosyl lactose, but the GOS that's commercially available, again, is not similar to what's in human milk. So I look at the fact that, we realized that human milk had more of these non-digestible carbohydrates. And again, my bias, my perspective is that the FOS and GOS were there as sort of a stopgap measure, to speak, that, we're trying to get closer to human milk. But again, a breastfed infant is not going to see these. So if we want to make formulas closer to breast milk, they shouldn't contain, particularly not something like fructooligosaccharides. You'll see on the commercial market that as some companies have added HMOs, they have removed these prebiotics from their formulas. Others have added the HMOs and slightly reduced these prebiotics. But again, those prebiotics have been shown to be safe, and they can have some effects on the microbiome modulation. But some of these other benefits that we've shown, for example, for the immune outcomes, are more specific to the HMOs. And so, again, I think, as James said, is if we're trying to get closer to human milk, the HMOs are going to be more reflective of human milk composition than having GOS or FOS.
Thanks, Sean. I also have some additional questions more around production and commercialization, and maybe this is also related to the previous question. And then this is for you, James. What are the criteria important for commercializing materials such as HMO?
Sorry, what do you mean by criteria?
What are the, I think, characteristics in terms of making HMO successful, maybe competitive in this infant formula market?
Okay. What we're finding is that this is a sort of balance between the sort of cost in use question that obviously a formulator would have in terms of how they prepare and design their product, versus the science that backs up the use of HMOs in that product. I think everybody in this industry is looking to get their product closer to breast milk. And as Sharon and I have said, HMOs are a way to get your product closer to breast milk. So I think there was a genuine desire to want to put these in. But that needs to be backed up by science, which is still emerging, and there's a lot of work going on, particularly being pushed through our academic program within dsm-firmenich, where we have many different academic groups around the world using our HMO library to help develop that science, as well as work that's going on internally as well. And I really encourage as many people as possible. I'm a passionate believer in HMOs, and I would love to see more people doing more science on HMOs so that we can bring the best to all the infants of the world. I think by doing that, as more people start to use HMOs in their products, we will start to see obviously volumes going up, and that will have a direct impact on prices, which will have a direct impact on how you can formulate at better levels in infant formula, but also across broader ranges of infant formulas. As I said in my presentation, HMOs are currently reasonably well represented in the top tier of infant formulas, and we're starting to see them come down in through the portfolio of companies. But the only way we're going to get them down to everybody and all formulas is by bringing the price and the science up. Price down, science up.
Okay, thanks, James. I have some scientific questions for you, Sharon. This is around the differences in terms of the birth as well as the maternal secretor status. I think to summarize, is there an effect different depending on the type of mom secretor versus non-secretor, or in the type of birth, the vaginal versus C-section, in the effect of an HMO?
Yeah, so there's been a number of studies that have looked at genetics, but also other factors such as route of delivery, maternal weight status, maternal diet. From the studies I've seen, there isn't an effect of how that mother delivers her infant on her HMO composition, but obviously how that infant's delivered affects its microbiome, and therefore those microbes might utilize the HMOs in the maternal milk a little bit differently. So for example, with C-section, it tends to take longer for the bifidobacteria to colonize. So again, most of the evidence we have is based on secretor status. And some of the early work actually done by Ardith Morrow and her group looked at this in Mexico because, as I mentioned, in Mexico, like other areas of Latin America, most of the moms are secretors. But because we have like this natural genetic experiment, they could compare babies suckling secretor moms versus non-secretor. And they found the babies whose mother was a non-secretor actually had higher levels of diarrheal diseases. So that was really, that first indication that 2FL and the other elongation products that are made from 2FL were conferring some benefit to the infant, even when they were being breastfed. So remember, breast milk has antibodies and lactoferrin and lots of, milk fat globule membrane that do protect against infection, but that lack of 2FL was having those babies at higher risk of diarrheal diseases, and particularly things like norovirus and other, as well as some bacterial pathogens. So, epidemiology, because moms do have different levels and different types of HMOs, can help to inform the science and the understanding of mechanisms. And then I think that's what then can feed the science of what HMOs we need to be making so that we can supplement them to infant formulas. So it's kind of a human milk informing, what we should be producing for infant formula.
Okay. Thank you, Sharon. Maybe in the interest of time, I'll start prioritizing questions. There's one question around the potential and thoughts around combining probiotics in an HMO. Perhaps I'll ask this to you both. And first with Sharon, is there a scientific view? What's your scientific view on combining probiotics in an HMO?
Yes, and I know we're getting short on time, so I'll just try to be succinct that There are some clinical trials that have combined probiotics with HMOs. Some of those were lactobacillus strains and some bifidobacteria strains. I think the strongest benefit that we know of now is actually combining that B. longum infantis with HMOs. And there are companies that are producing the B. longum infantis as being used in clinical settings. But if you give B. infantis to a formula-fed infant without HMOs, it actually doesn't colonize. That baby needs to either be breastfed at the same time or be receiving formula with HMOs. So that's a perfect example of a true symbiotic where you're providing the fuel and the bacteria and making the magic, so to speak, as they are interacting. And I think the clinical studies that I showed you were suggesting, why that might be happening and be important mechanistically.
Thank you. And then James, maybe from industry and commercial perspective, what's your view in combining probiotics and HMO?
Yeah, I have to say this is an extremely exciting area, and I think we need to be, really aware that this is also emerging science. But I actually see this as one of the next thresholds in HMO development, is how the symbiosis with particular probiotics actually works, and what benefit that that can give to the infant. What we're finding in our own internal work is that some probiotics work a lot better than others. And some HMOs work a lot better than others, and combinations of HMOs work a lot better than others. And that is extremely interesting, and really got our curiosity running in terms of how we can, formulate, or how we can work out the best combination of these materials in order to give the best benefit to the baby. So I think for now, there's not much more I can say, but very much watch this space. But this is something we're extremely excited about.
Okay. Excellent. Maybe we have time for one more short questions. Let me try to find one. So I'll ask this to you, Sharon. What's your view on the balance between the transient and long-term establishing bacteria in the gut?
Oh, boy. Wow. So I think that we know that it's important to get the bifidobacteria established in that human milk, and human milk oligosaccharides support that. And I think that early exposure to the microbiome, even if we find that bifidobacteria tends to go down over life, is really important for that host microbe interactions and training the immune system. So, sometimes we'll say, oh, well, those bacteria aren't there anymore. But I think that they were there at the right time and in the right place to be educating that infant. And certainly bifidobacteria and immune development and the microbiome mediation of that with HMOs is critically important. So I think that if we can support that process, as I mentioned, C-section babies tend to get bifido a little bit later and different strains, and breast formula-fed infants have bifido, but they don't have the B. longum infantis, and that's what HMOs can help us to promote. So when we look at bacteria, it's important to think about who's there, when they're there, and particularly what strains are there.
Thank again both the speakers, James and then Sharon. And thank you for the audience for active participation and throwing some challenging and critical questions too. This concludes our webinar for today. The recording will be available to you, and it will be communicated separately. Thank you very much once again to everyone, and have a great rest of the day. Goodbye.
Thank you.